Hi, my name is Koji Ishiya, and from Kansas University. And thank you for time to view my poster. My poster title is Human Biological Sex Determination using XY linked K-mer signatures. In this presentation, I will introduce for determining human biological sex. First, I will explain the background of these studies. The biological sex determination in humans can be broadly categorized as morphological method and molecular science. In the morphological method, sex determination mainly use features with sexual dimorphism, such as previous site. However, it is challenging to determine sex in individuals with significant bone loss or immature juvenile remains. And additionally, these methods are generally applied to degraded samples. The DNA-based determination targets genes or different lengths such as the amyloid genes on the sex chromosome. By detecting difference in the PCR amplified fragment lengths. However, this method may not work with degraded archaeological sample where a phenomenon called early dropouts and this occurs leading to the failure of detect expected amyloid genes, thus preventing and aware of accurate sex determination. In response to these technical challenges, method using mixed generation sequence NGS have been proposed. This method called calculates the ratio between the number of sequence read aligned to X and Y chromosome to determine the biological sex. This approach is mainly applied to ancient samples extracted from archaeological sites. However, these have several technical issues with this method. The first issue is that the range of RY is ambitious, making it difficult to determine sex depending on sample condition. And the Y chromosome in particular contain many repeated sequences, and in some samples contaminated with DNA from other species. The RY values can vary significantly. Moreover, when the initial number of the read is low, the RY values can regulate generally greatly, making accurate sex determination challenge. The second issue is difficult in accessing the scale between the male and female. In mixed samples, in the forensic cases, the probabilistic representation becomes crucial. And in this study, I developed a new method called Onyx to address these above technical issues. Onyx detected K-mers specific to the X and Y sex chromosome uses a ratio of the K-mers for sex determination. These K-mers are unique sequences expected to appear once on the X or Y chromosome and show raw homology with autosomal and external other species genomes. When the K-mer derived from the X chromosome are referred to the X as KY, and those from X chromosome as KX. And this method detected KY and KX from next generation sequence and calculated the RKY, and this values is ratio KX and KY. And then this method constructed a logistic model and based on the RKY values to the probabilistically determined sex. And I evaluated effectiveness of Onyx using over 2,500 males and female samples from the 100 Genomes project. The result shows these methods achieved a UC over the points 97 indicated an extremely high accuracy in sex determination, and for more validation compared to existing methods, and using for genome sequence at 5-fold coverage demonstrated our methods could determine in about 1/4 of the time compared to existing methods. And additionally, the RKY values remained almost unchanged even with extremely low coverage data below 1/4 coverage. And this suggested the method is robust even where applied to low coverage generators such as degraded archaeological samples. Overall, this method demonstrated significant reduction in processing time to compare to existing methods and provided a stable sex determination even at a low coverage, which contributed to more efficient DNA-based human biological sex determination. And that's concludes my presentation. Thank you for your attention.